Good day, Honors Biology students. Welcome to video lecture episode 23. Today we're going to continue our discussion in, remember, section 3 of chapter 12. Remember, once again, chapter 12 is titled Molecular Genetics, and in section 3 we're talking about DNA, RNA, and protein, the process in which, of course, genetic information is passed on. And last time, when we finished our discussion, we had just gotten into talk about what is referred to as the central dogma of biology, that being the flow of genetic information going from DNA to RNA to protein. So now we're going to move to slide 11 and we're going to start breaking down that central dogma a little more. We're going to start with RNA. RNA, like DNA, is a nucleic acid. There are at least three ways RNA is different from DNA. One, RNA is single-stranded as opposed to DNA being double-stranded. Two, RNA has the sugar ribose where DNA had deoxyribose, and third, it has the nitrogenous base uracil, which replaces thymine in binding with adenine. And if you look on slide 11, you can see, as well as slide 12, you can see the image where just comparing, graphically speaking, DNA and RNA. All right, now let's move a little bit further forward. Now, there are three major types of RNA found in living things, and that is mRNA, which is messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. Messenger RNA is long strands of RNA nucleotides that are formed complementary to each one of the DNA strands. All right. Transfer RNA are smaller segments of RNA nucleotides which transport amino acids to the ribosome, and ribosomal RNA is the type of RNA that associates with the proteins to form ribosomes in the cytoplasm itself. Now, if you look on slide 16, you can see a graphic representation showing all those types of RNA. If you look on it, there's a green one that looks almost like it's got little teeth mark, and it's got U, G, G, A, 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 G, A, G. That one is the messenger RNA. The little yellow rectangle coming in and connecting, that would be considered the tRNA. And if you notice above that, you see the polypeptide chain growing up it, which has the amino acids. And then also there's one other thing that I want to mention as well on this image, and that's the RNA. The rRNA, and that's the ribosome itself, which is holding together both the tRNA and the messenger RNA together in this process of what's called peptide synthesis. Now, if you look at table 12.2, which is on slide 17, you can see how it compares both the structures and the functions of the three types of RNA. Okay, so just take a look at that, kind of review that, make sure you understand that. If you have any questions, let me know. All right, so let's move to slide 18 and let's talk about transcription. Now, the central dogma, the flow of genetic information, is divided into two stages, and those two stages are transcription and translation. We're going to start off with transcription first. All right. Transcription involves the synthesis of messenger RNA from DNA. During this process, the DNA code is transferred into messenger RNA in the nucleus. Once the DNA code is transferred into the messenger RNA, it then leaves the nucleus and enters the cytoplasm itself. The messenger RNA travels to the cytoplasm for protein synthesis. Now, as we continue describing transcription and then translation, look at figure 12.13, which is on slide 20, because it's going to help you. And also, if you could, illustrate this in your notes so that you have something to connect visually with what we go over. So let's continue on. Now that we have described transcription in an overview sense, let us proceed to explain some of the particulars of that process. The DNA is unzipped while in the nucleus, and the enzyme RNA polymerase continues along to regulate RNA synthesis. Remember, synthesis is to make. Now, if you look at slide 22, you can see a graphic representation there of how the RNA polymerase is unwinding the DNA and getting the messenger RNA. Remember, this is the process of copying it. This is how it begins. It has to actually unwind it so it can make the copy that it needs to make. Now, the enzyme binds to a specific section where it will synthesize messenger RNA, so it doesn't just random, it picks a particular spot. Now, as the DNA unwinds, RNA polymerase initiates messenger RNA synthesis in the 3' to 5' direction. So if you see on the image on slide, I believe it's 23, it looks like a 5 with like a little comma above it, that would be representing prime. So remember, the direction is going from 3' to 5'. All right, now let's go ahead and close on slide, I believe it is slide 24. The section which the RNA polymerase is reading is what we call the template strand. The messenger RNA strand, which is made, is what we call the complementary to that template strand. Now, after the messenger RNA strand is made, it exits the nucleus via nuclear pores. All right, ladies and gentlemen, I do hope this lecture has been helpful. If you have any questions, you can leave them in the comments below. Send me an email or message. Have a nice day. Take care. Goodbye.